Okay. So hi everyone again. We're here to welcome Erica Griffith. She's a MD PhD student at SUNY Downstate with Bill Lytton, and she's also working at the Nathan Klein Institute for Psychiatric Disease with Sam Nimatin on this model of the auditory thalamocortical system, which I'm also been working with her, and she's been doing an amazing job with the model. We've made a lot of progress, and I'm very excited to see her talk. So please welcome everybody, Erica Griffith. Thanks.
Okay, thank you very much, Salvador, for that intro. So hey everybody, I'm Erica Griffith. You heard me give a talk last week, and I believe we did some exercises with a group of you last week as well. So today I'll be taking you through some more information about one of the projects here in the lab. And throughout the course, you've heard it mentioned here and there. So hopefully today we'll kind of round out more of the picture. So for today's talk, I'll be talking to you about a data-driven model of the auditory thalamocortical system. So first, to go over a little background to give you some context. So this model is part of a broader effort to investigate the neuronal oscillations that are observed in the auditory cortex during speech processing. And so since this is about speech processing, let's first talk a little bit about speech. So here is an example of a speech waveform. So along the bottom axis here is time. Along the vertical axis would be sound pressure, usually represented in decibels. And now speech generally has a quasi-rhythmic waveform, which we can see in this example here. So we can keep that in mind for later. Signal can also be represented as a spectrogram. So here time is still on the x-axis, but on the y-axis, instead of sound pressure or decibels, is frequency, hertz. So the grayscale intensity, sometimes it's color intensity, but so the color, as it were, generally represents the amplitude of the frequency at that particular time point. So say at time point, so say let's say we want to look at this particular frequency band, for example. So say this is what's happening in the 780 to 930 hertz range or something. And so in other words, again, this would be 780 to 930 hertz here on the y-axis. We have time on the x-axis, and grayscale corresponds to the signal intensity. Let me know if there are any questions on that. So that kind of brings us to the first major concept that we need to understand. And that's that a sound signal can be filtered or broken down into activity in different frequency bands. In auditory terms, these are sometimes referred to as critical bands. Meaning within a certain frequency range, it's hard for us to distinguish perceptually between two different tones. So you can kind of think of it like a unit of auditory resolution, if you will. And the reason that this is important is because that's what our cochlea actually does. So in this context, it acts as kind of a series of bandpass filters, which literally means that only a certain frequency band is going to pass through each filter. So in other words, the cochlea can take an incoming waveform like this, and each part of the cochlea will extract the contribution of its frequency range from the entire waveform. So let's say it's this frequency range, it'll extract just that information out. And we can also note that this filtered waveform, broadly speaking, has two kind of components to it. It has the fine structure in here, and it also has an envelope, which is the smooth curve that outlines the edge of an oscillating signal. Now, this envelope is the important part for us. So according to a growing consensus, this envelope here, and this isn't exactly what it is. There's a little bit more nuance to this, but so if anybody's interested, wants some papers, I'm happy to share. But for now, we'll explain it as that this envelope is the feature that's getting tracked by the oscillations in our cortex. So in other words, oscillations that are occurring in the auditory cortex start to phase lock to this envelope when there's a quasi-rhythmic kind of incoming auditory stimuli. So we can take a closer look at the structure of this envelope. So again, we have here a sound waveform. And again, the envelope here is just the outline of that waveform. We won't worry about the filtering aspect for now. We'll just focus on the periodicity of this envelope. This is a figure from just a paper where they were looking at syllables. So we can see, so we can see from here that a kind of syllabic rate that occurs: drive, round, pick, turns out that this periodicity, this rate of syllabic information, it's pretty stable at 4 to 7 hertz, and that's true even across languages. So we can see here a figure from a 2016 linguistics paper that's on this very phenomenon, shows that in all these different languages, the rate of incoming syllabic information clusters in this same range. So why is this important? So now we can go back to the figure that we saw earlier on that first slide. And this actually shows real data from our macaques. These are wavelet spectrograms taken from macaque A1. I believe this is in layers 2, 3. So we can see oscillations that are occurring at different frequencies. We have the delta at the lowest end, we have theta, we've got alpha, beta, and on the higher end we have gamma, and then the high gamma. So recall that the syllabic information comes in at around 4 to 7 hertz. Now, going to looking at these cortical oscillations, we can see that the delta and the theta bands here occupy this very same frequency range. So kind of in the 4 to 7 hertz range. And now this leads to something that we call entrainment. And that is where the phase of the oscillations in cortex, typically in this delta theta range, they align themselves with the phase of the auditory signal envelope. And we do have reason to believe that this isn't just an artifact, this isn't just an epiphenomenon, and entrainment might be a necessary process that's underlying speech comprehension. So there's, at this point, been a number of different studies that found that disrupting entrainment, so disrupting the ability of oscillations to phase lock to the stimulus, whether that's using transcranial magnetic stimulation or lesions, things like that, that impairs speech comprehension. And so when theta band activity does not follow the speech envelope, intelligibility does degrade. So here's kind of a cartoon schematic of this, just so you can drive a point home. So on the left here, we have some cartoon neuronal oscillations. These are occurring in a low frequency bands like the delta theta band, less than 9 hertz. And then we have the onset of this quasi-rhythmic auditory stimulus, like a speech signal. And we see that when this signal starts up, we get an alignment of the phase of these cortical oscillations with the phase of this incoming auditory stimulus. And again, we do think that this has functional implications. There's the behavioral studies and things like that. So one of the hypotheses about why entrainment is helpful or even necessary for comprehension is that maybe this phenomenon might allow us to parse continuous streams of speech into syllabic chunks for further processing. And there might also be an interplay with attention, and that this kind of phase locking helps to sort of block out other competing misaligned streams of speech, things like that. You can Google the cocktail party phenomenon for more information on that. And to kind of briefly conceptualize this, we can turn to this diagram. So let's look at this figure. So the blue waves, did somebody...
Yeah, is there an alternative hypothesis, Erica? How could it not be critical for speech comprehension?
That's a good question, Bill. Let's see. So, I mean, there is the idea that maybe it is just it is an epiphenomenon, that it's not actually that it's something else that we're capturing, with these oscillations. That there's some other processes underlying this that is actually doing it. So
well, a feed-forward process can't seem to possibly explain a dynamic frequency-based comprehension. But that's, yeah, I have never understood what the alternative is. Sorry, go on.
I know that there are people who refute this, and I'm trying to remember the name and the arguments of that guy. I can look that up later if anybody's interested.
Yeah,
I do have that bookmarked, so it is an interesting question. So let's see, where am I? Okay, so we can turn to this diagram here. Again, just kind of a cartoon depiction of what's going on or what might be going on. So here the blue waves indicate the cortical oscillations, and the red blocks are kind of standing in for our quasi-rhythmic stimulus that's coming in. And the thing to note here is that the phase of the cortical oscillations has been correlated to periods of low and high neuronal excitability, which makes sense. Oops, sorry. So this bottom panel here shows kind of optimal phase alignment, where information from the stimulus is aligned with these periods of high excitability as indicated by the cortical oscillations. So since the signal's coming in at a period of high excitability, the information is able to propagate through the network and be a candidate for further processing that might be necessary for speech comprehension. And the top panel here shows kind of a suboptimal phase alignment, or misalignment, if you will, where the information that's coming in from the stimulus is coming in at periods of low excitability. So that means the information may not get efficiently propagated, if at all. So, this is kind of the idea behind the cocktail party phenomenon. Might have to do with attention. Say your cortical oscillations are locked onto a stream of speech that your friend is producing. So if you guys are locked, it's going to look like the bottom panel. But as we may distantly recall, in the past at parties, there were multiple people talking at the same time. So this top panel might represent more like what's happening between your brain, and the person in the group next to you, something like that. So basically we've covered, that entrainment occurs. We've covered why it might occur in terms of the functional implications. But what we are mostly concerned with for this project is how entrainment occurs. And so for this, we need to look at the thalamocortical circuit. So, so here we know we have the auditory cortex in the circuit, considering everything we've just been talking about. With it, we have 6 layers here, layers 1, 2, 3, 4, 5, 6. There's also the thalamus. And so for now, we're going to focus on the medial geniculate body because we know that this is a major source of inputs from the thalamus to the auditory cortex. We also do have thalamic reticular nucleus modeled, and we can talk about that in some later slides. Now, there's a lot of literature out there suggesting that there's 2 distinct sort of pathways or populations of neurons in sensory thalamic nuclei, and they're called the matrix pathway and then the core. Now, so these 2 populations are distinguishable histologically, as well as anatomically in terms of where they project. So the core neurons project to layer 4 of the auditory cortex. It's thought that they're taking, the auditory information, just the, regardless of what it is, just the whole data dump sort of, and transmitting it directly to layer 4 in a tonatomic fashion. So the matrix neurons, on the other hand, seem to have much more diffuse or sort of non-specific projections to interneurons in these supragranular layers. So there's 1, 2, 3. And these interneurons inhibit pyramidal neurons in layers 2, 3. So now that we have this background, we can take a closer look at the circuitry. So again, we start with the core neurons, giving a stimulus-driven spike train into layer 4 of A1. The signal then propagates upwards from layer 4 to layer 2, 3. These matrix neurons are going to activate supragranular inhibitory interneurons, which then might inhibit pyramidal neurons in layers 2, 3. So that allows these inhibitory neurons to sculpt the incoming signal from layer 4. So if you notice, this is very similar to the this is very similar to the figure that we showed earlier. So you can see that these kind of rhythmic periods of high and low excitability, they could sculpt the incoming signal into something that looks like this. So just to summarize again, so we've gone over, the speech is quasi-rhythmic. We have syllabic information coming in at a rate of 4 to 7 hertz. We know that entrainment is when low frequency oscillations in the auditory cortex sort of phase lock to the stimulus envelope, at the rate of incoming syllabic information. And we also see that this entrainment appears to be functional, not just an epiphenomenon. And sort of our main hypothesis, and that's that matrix, or that medial geniculate body neurons in the matrix pathway and supragranular interneurons in the cortex are a key part of the circuitry that allows for this entrainment or this phase locking to occur. So any questions so far before I go on and talk about our approach kind of as a, okay. So this is kind of a summary of our approach strategy. These aspects all kind of interact to support each other. So we have the in silico modeling here, and that's informed by the empirical work. The modeling is then ideally used to make predictions about what might be going on in this circuit, in this brain region. And those predictions can then go back and be tested in living systems. So, primarily we are, okay, I should mention that this, we do have a lot of data. We have a robust collaboration with a group at the Nathan Klein Institute, Peter Lakatos' group are working with these macaques, and they're collecting a lot of data, current source density in A1. They even have some human EEG and ECOG data. There's single unit recordings, so there's data taken at rest, data taken during speech tasks. And these all help us understand, what oscillations are present at rest, how these oscillations change in response to stimuli, and how the entrainment described earlier correlates with behavior and perception. So these are the types of questions that this kind of data can examine. And so this data is also useful, of course, for building and also validating a biophysically detailed model of the thalamocortical system, which is what this course has been about. So briefly, the idea is we can use this model to generate predictions about the mechanisms underlying the entrainment phenomenon, because we're going to have a more kind of accessible, granular view of all the pulleys and levers in the system. I'm sure you guys have gotten a feel for that, as you've gone through and built things and done the tutorials and exercises in NetPyNE. You can much more easily, lesion things by setting connection weights to 0, things like that, things that would be much more difficult, in a living system. So then we can kind of ping pong these predictions, things like this. We can ping pong these predictions back to the in vivo side of things. And that's where experiments, say with, targeted neuromodulation, other things like that, can be used to test these predictions. So we've got this kind of loop going on here. So now we've talked about the background of this project, our approach to this, and any questions on that so far? Alrighty. And so from here, I'll summarize the two things that we want to show. So this first one is that cortical oscillations in the delta theta range will synchronize to naturalistic quasi-rhythmic auditory stimuli, as we've been talking about. And again, as we've been talking about, we also want to see whether or not entrainment relies on this oscillatory phase reset mechanism orchestrated by the thalamic matrix pathway and by supragranular interneurons and cortex. And we want to show these things both in vivo and in silico, kind of in line with the approach strategy that we just talked about a couple slides ago. So let's talk about the first point for a bit, how we want to show that cortical oscillations synchronize to quasi-rhythmic stimuli. And first, we'll touch on the in vivo context of this. Okay, so first thing to note here is that in order to show synchronization of cortical oscillations, we have to be able to detect these oscillations in the first place. So to that end, Sam Niemoten, who's one of the PIs on this project over at the Nathan Klein Institute, has developed some software to accomplish this and detail some of its capabilities in a manuscript called The Taxonomy of Neural Oscillation Events in Primate Auditory Cortex. And this title here is a link if anybody is interested in reading about this further. So again, the software can detect neural oscillation events in electrophysiology recordings. I can see an example down here. You can look at event-related potentials in A1 in the different layers. And here, this is kind of a cataloging different features of these oscillations, so the number of cycles, things like that. So this is kind of how we're cataloging and analyzing the non-human primate data. And as I'll mention later, the same software is what's being used also to analyze these things in the model. So that's called the O-Event package. And again, feel free to go to this link if you are interested in looking and learning more about that. So next, we can talk about this from the in silico angle, which has been the main focus of what we've been doing in the course for the past week or so. And we're going to talk about how we can look at this phenomenon from a modeling perspective. So to this end, there's kind of 3 steps that we need to take. So the first is, of course, going to be to construct a biophysically detailed, model of this pathway. And the next is going to be to tune that model, which I'll talk about what that means and how we do that. And the next will be to validate that model using empirical data that we have from our macaques. And so now we'll kind of go through what each of these means. So the first thing we're going to do is talk about constructing the model. And this is something that you've heard us talk about in previous lectures, say on building multi-scale models. We've had other exercises that have touched on pieces of this and things like that. And as you all know, this modeling is being done with NetPyNE, using all the coding and organization techniques that we've been talking about. So let's just go through the pathway that we've modeled here. So first, we'll start with the peripheral structures like cochlea, inferior colliculus. And so for these, we've used so-called phenomenological models. And these are different than the biophysically detailed models that we've been discussing throughout the course. Phenomenological models, they capture the signal transformations that are occurring in these areas. So the signal manipulation that's occurring is very data-driven, it strives for accuracy, but the way that those signal transformations are arrived at doesn't necessarily involve the kinds of conductance-based cell model interactions and stuff that we've been discussing. So these models produce outputs from inferior colliculus that are used to drive the thalamocortical cells in the downstream, more detailed, more biologically realistic portion of the auditory pathway model, which I'll be talking about next. So to give you a little detail, the cochlea, as we discussed earlier, it filters sound into different frequency bands. It transduces that signal into action potentials. The auditory nerve transmits those transduced signals. And it does exhibit several nonlinear response properties, rate saturation, frequency adaptation. The model that we used, and I'll include a link to it, to a publication about that model, does aim to capture those properties. So similar to, here's a link up here if anyone wants to read about this model. So similar for inferior colliculus, there's definitely a bunch of nonlinear things that happen there as well, amplitude and spectral modulation tuning, things like that. So basically, we have, cells in the inferior colliculus. We have this signal that's being filtered, going up auditory nerve, arriving at inferior colliculus, and then different parts of the incoming sound signal are modulated according to its amplitude and frequency components. And if anyone wants more, information or citations on the details of this, if you're interested in, in this pathway, feel free to message me. I'm happy to provide, a bunch of citations, and that's kind of a whole interesting side quest in and of itself. So we use these phenomenological models to capture these processes, and these models, in particular from this group, are great because they mitigate some issues that are seen with other phenomenological models that don't kind of include these, don't really capture these characteristic nonlinearities that I kind of mentioned. So you get, kind of a breakdown in higher frequencies or higher sound levels. So using these models means that we have, inferior colliculus outputs that are really useful through a broad range of frequencies and noise and things like that. So what's great about this setup here is that having these peripheral models allows us to provide our pathway, our model, with arbitrary sound input. And the great thing about this is it enables us to use matching sound stimuli. We can present the exact same stimuli to our macaques and to the model. And that gives us a really good baseline for kind of determining the validity of the model's behavior in response to these sounds. I can just give you a little.
Would you like a banana, Peter?
So that's just an example of one of the typical stimuli that we might be giving to the macaques and to the model as well. Okay, great. So now we can move on to the more kind of biophysically detailed parts of the model that are, using NetPyNE and conductance-based cell models like we've been talking about. So the next part of the pathway here is the auditory thalamus. Now this, again, we've modeled using the kinds of cell models and things that we've been talking about throughout the course. And the thalamic regions that we modeled include the medial geniculate body and the thalamic reticular nucleus. These populations are divided between the core and the matrix pathways, as we mentioned earlier. So for the medial geniculate, oh, sorry, this is actually behind here. Shouldn't be. Well, apologies. Okay, so we have core pathways, we have matrix populations as well. And similarly, we have the thalamic reticular nucleus, core, and matrix. Apologies for that figure mishap. So as we mentioned, this is the kind of information you want to get from the literature in terms of, what cell types are participating in the circuitry that you're interested in, what their density is in this location. For example, we're just modeling a certain portion of the auditory cortex, so just, one column. So you have to figure out, kind of the ratios of cells in this column to cells in these thalamic regions, because you're not going to model the whole thalamus. You're just going to model kind of a part that corresponds to a single column in A1. So you want to go hunting around in the literature for that kind of information. And, it's also very important, as I mentioned in the multi-scale model building talk, to think about, what animal model that you're looking at. For example, in this, we know that in primate versus rodent, there's pretty big differences in the numbers of local thalamic interneurons. So getting that kind of information can be very important, kind of harkening back to our our multi-scale modeling talk. Let's see. So this is also where you want to be looking at kind of the information about different dynamics of your particular cell types. So cells in this region tend to have very interesting bursting properties, which is something we wanted to capture as well. If any of you in the course have studied phenomena like absence seizures or slow wave sleep, then you'll know that the bursting capabilities of thalamic cells can play a very important role in the oscillatory dynamics that arise in those situations. So we wanted to be able to capture that here. So to illustrate the proposed circuitry, is that a question?
Yes, I have actually two questions. If you could go back maybe a little bit where you have the all the connection back. Yeah, exactly here. I have two questions. So the first one is, so you said that when hearing any kind of sound, we have a transduction that will be translated then in a series of action potentials. Okay, yeah. So it's like, this is just out of curiosity, it's like that increasing the, I don't know, the volume of the sound or the frequency of the sound, you have an increase in the number of action potential. There is this kind of, let's say, correspondence between the two, or it's, I don't know, different, like, I don't know, specific frequency band correspond to a lower number of action potential and so on and so forth.
Yeah, so that's a very interesting question. And, if you're interested in those types of things, there's, I know Kendall has a great chapter in the, what's it called, Principles of Neuroscience about this. And there's a lot of different interesting publications that talk about what you're asking. So generally, the cochlea, like we mentioned, you're going to have different, the cochlea is a sort of a structure like this. It's stiffer at one end, looser at the other. So different parts are going to vibrate sort of to specific frequency bands. And so the frequency doesn't necessarily, the frequency range doesn't necessarily correspond to action potential frequency. Volume can, but then, once you turn those into action potentials, they'll start going up the pathway. It gets a little bit more complicated. So yeah, intensity of the sound signal, like volume, can cause oftentimes an increase in the rate. Frequency, not as much, because you're kind of going by section of the cochlea, so that's encoded slightly differently. But again, like I mentioned, there's also these sort of non-linearities that occur with the cochlea, with the auditory nerve signaling, where you might get rate saturation. So at a certain point, above a certain volume, you might not get an increase in rate and things like that. So, so there's definitely a lot of very interesting sort of complex signal processing stuff that occurs, as you go further and further up the pathway. That's it. But it is a good question. I'm happy to send you citations and chapters and stuff if you're interested in, kind of learning more about that.
Yeah, that would be great. Yeah, this would be really interesting. Thanks. And the second question is about, so I see here in this scheme, let's say that from one side, so from one ear, let's say, we have with these red arrows, you have the connection to both sides of the brain, let's say the right and the left hemisphere. So it's like the same amount, let's say, with information on both sides, or would be, I don't know, because I'm thinking in terms, for example, of the visual system, where you have basically the optic tract crosses, and then you have that the highest information on one side comes from the contralateral side, so from the contralateral eye. And I was thinking if something similar occurs also here in the auditory system.
So there is, so yeah, that's also a very good question. There is decussation, or crossover of pathways in the auditory pathway as well, particularly in midbrain around the level of the cranial nuclei. But so actually, what's interesting about that is there's delay actually. So if you think about sound coming in that's directly in front of you versus a sound, let's say, that's rotated 30 degrees, but at the same kind of horizontal plane. The one that's rotated 30 degrees, that's going to hit one of your ears, say your right ear, just a little bit faster, just a little bit before it hits your left ear. And so your brain, the way that it kind of encodes that is by this delay across, because you have slightly longer axon going from, here to over here. And so that's kind of how your brain localizes where the sound is coming from in this plane. So there is redundant information going in from, both to ipsilateral and the contralateral side. And that is used actually to kind of localize the sound in space. So that is, among other things, but that is an interesting question. There is crossover, there is redundancy. It's a little bit different than in the visual cortex, if I recall correctly. And again, I'm happy to send, I think Candel does a pretty good job of describing this, and I can send you the page numbers and stuff, because I know I have that written down somewhere.
Yeah, okay, thanks a lot.
Yeah, no problem. Alrighty, any other questions on this? Okay, let's see. So, okay, so yeah, I just wanted to illustrate, a little bit more of the proposed circuitry. Show you a little schematic that'll hopefully illustrate some of the thalamic stuff that we were talking about. So we wanted to make a working model of this circuit. So we have the reticular nucleus here, medial geniculate body here with our excitatory thalamocortical cells, and we have our interneurons, which are inhibitory. And these are local inhibitory interneurons that go ahead and inhibit the excitatory thalamocortical cells. We have A1 over here. So let's say we have excitatory input coming from A1, or going from A1 into the reticular nucleus, which some of you may know, some of you may not, that the reticular nucleus sends inhibitory projections. So this goes ahead and it inhibits, say, an interneuron that's in the medial geniculate body. This disinhibits this thalamocortical cell, which then can burst fire, send it bursts to A1. And the reason that it does this, again, this goes into the specifics of thalamocortical cell dynamics, which is why, it's important to get all that information from the literature, or electro, all those things we talked about, is because this was being held at a hyperpolarized potential by this interneuron, it is de-inactivated. So that means that it's being held, and once it's released from that, slightly hyperpolarized state by the release of this interneuron, then it causes T-type channel activation, and we get this bursting. And so then we have this hypothesis here that perhaps let's take out this. Let's keep everything here. Assume we're in the same state where this hyperpolarizing influence from the local interneuron has been released for a bit, and then picking up where we left off. The cell has been bursting for a bit, so now its T-type channels are going to be inactivated again. And then, say at this point, we send some ascending input into the thalamocortical cells from earlier in the pathway, and then at this point, the cell can't burst anymore because of its inactivated T-type channels, so it transitions to tonic firing instead. And so this is kind of one of the hypothesized mechanisms by which we might get sort of a break or a reset in what these thalamocortical cells are doing. So I just wanted to kind of include this. Let me know again, there's some papers by Edward Jones and things like that that go more into the circuitry and how this might all tie together. So, so that kind of dips into the thalamocortical or the thalamic part of the pathway. And next we can talk some more about the cortical column in A1. And you guys have heard us mention this, a lot over the past week or so, especially again in the multi-scale modeling talk. So if anybody is curious about kind of abstractly how to apply some of the stuff I say here to your projects or other things, feel free to look back at that talk. I included the PDFs, the slides in the Google Drive. So I'll just go over a little more about this now. So here we have on the left, we can see this column has many neurons, many millions of synapses. And here we can see this column, kind of a general schematic of it here. We have the geometry, which we talked about last week. And here we have the relative normalized layer boundaries that we got from our collaborators at our macaque collaborators. We talked about how to instantiate those. We also have our densities of the neurons, per layers, and then, this is kind of the overall density per layer. And then we used things like these figures, these things from the literature, as I mentioned, to kind of break down by cell type, what the density of each particular cell type within a particular layer might be. This is about interneurons, kind of laminar distribution of interneurons in cortex. And so then, of course, there are the actual cell populations. So again, I touched on this as well during our multi-scale modeling talk. And again, the important things are the identity of the cell types that are involved. For example, Here in layer 1, a lot of models don't include much at all up in this layer. For a long time, we didn't think there were a lot of cell bodies up here, but we now do have data supporting the existence of these neurogliaform interneurons in layer 1 in macaques. And so we included them here, and we did our best to model their properties based on the literature we were able to find. This is, again, the kind of thing that I mentioned in the modeling talk. And you may also recall that our circuitry hypothesis relies on connections from thalamus to the supragranular interneurons, including layer 1. So, the cell population was critical to include for us. And just to go over some more stuff about the cells. So here are some firing rates from models of several of our other cell types, firing rate curves, excuse me. And I mentioned in that talk as well that this can be an important thing to kind of find and replicate because it can, it's an important part of how cells are going to be transmitting information to other parts of the pathway. And here are some example traces from an excitatory cell model that was used in the cortical column as well. And of course, one of the other major things that we touched on that we need to figure out is the connectivity between all of these populations. So not only the cortical populations, but the thalamic ones as well. And so for this, as we discussed, we combed the literature to come up with a data-driven connectivity scheme that includes the probability and the weights of all the connections between the cell types outlined here. This is just an example of a connectivity matrix between the different cell types. Again, this is something you can generate with NetPyNE if you want to visualize it. And we also worked, like I mentioned, on the subcellular connectivity as well. Again, using data, driven by data that's been reported for sensory cortex in the literature, citation down here. And if anyone wants more details on, how this is coded and, how to apply this to your projects and stuff, feel free to refer to the slides from the talk. And I'm also happy to talk, about the details of your particular, any particular problems you are having as well. So just kind of putting this all together, we have this sound that's coming in. We have these phenomenological models for the peripheral structures up to inferior colliculus, and the inferior colliculus is generating these outputs that are then being used to drive the thalamocortical populations in the auditory thalamus, including the thalamicorticular nucleus, medial geniculate body. That's being connected up, in a data-driven way to the primary auditory cortex. All of these use data-driven cell models, data-driven connectivity, and we have this overall schematic that kind of just puts it all together. So that's kind of the structure of the entire pathway, kind of a brief synopsis of each of the parts of it. Let me know if anybody has any questions on that. Alrighty, so next we come to, so you might recall that there's a couple more steps that come after this. So that's constructing the model. No trivial feat for sure. And next we come to tuning the model. So the objective here is to tune the model such that, and validating the model. So we'll talk about tuning first. So the objective here is to tune the model such that, at rest, so in a spontaneous condition, so to speak, not being given any speech stimuli, something like that, that we meet 3 criteria. So ecologically valid firing rates for all the categories of cells in the model. We don't want to see, just like runaway excitation. And we want to see activity at rest that, generally matches up with activity that we see experimentally. So, what does this mean? What does it entail? So it requires a few steps. So first, each synapse is independently provided with just background stimulation. We want to see that the frequency spectra for the interspike intervals of the background inputs is flat. So basically, we're feeding it just sort of this white noise spectra. And this should take place before activating the connections between the cells. So all the data-driven connectivity and stuff, this should happen before we activate all that in order to give us sort of a clearer picture of the origin of activity in our different cell types. Because once we add in the connectivity data, it can get a little more difficult to, ascertain why a cell population is spiking too much or too little or something. So we might want to look at cell population activity without that connectivity involved. You get sort of a baseline. And what we want to see from our cells are low levels of spiking activity, so around 1 to 5 hertz. So this kind of background input shouldn't be enough to, for consistent high rates of spiking. So just an example of what that might look like. We have some voltage traces from different cell types in the model in response to this noisy, background input. We've got a somatostatin cell in layer 2, firing rate around 5, PT cell, layer 5B, firing rate of 1 hertz. So that's the kind of thing that you want to be looking for. And so next we activate the cell connections and go in and adjust the so-called the network level connectivity parameters. And so these are things, for example, like the synaptic gain, which is a commonly used construct, and can be, for example, so say the gain from point. So it's between, so these postsynaptic potentials, sorry, let me explain this. So between an inhibitory and an excitatory cell, you might have the weight of this connection. And, you know, we also, what we're talking about here when I say gain is sort of the gain in the postsynaptic potential on this, you know, excitatory to inhibitory connection. And I want to note that these parameters are, you know, they have a very coarse level of biological detail. So we're not talking about necessarily specific cell types and things like that. These are parameters that are going to affect lots of different types of synapses at once. So like a chunk of excitatory to inhibitory synapses. And this coarseness allows us some degree of freedom when tuning them. We do try to constrain the parameters to ranges that are biologically realistic. You know, we don't let the gains get so high or so low that the postsynaptic potential voltages are, you know, totally biologically unrealistic. But you know, as you guys are starting to see from the multi-scale modeling talks and the different exercises and things like that, we don't always have, you know, complete data all the way through from cell model to connectivity, you know, to density and stuff that is all the same animal model, you know, all the right cell types, everything like that, all the right brain region. And so sometimes there are adjustments and things that need to be made. And so, you know, having this sort of, having these dials that are a little bit more coarse does help to, you know, tune the model such that it is behaving in a biologically realistic way, you know, considering that we have to make compromises along the way as we're constructing the model. So we activate all the cell connections, and then we adjust these sort of gain parameters until we see that the cell types in the model are firing, you know, at ecologically valid rates. Again, there's no runaway excitation, you know, which could, for example, could mean there's too much excitatory to excitatory gain. And I want to note this is a very, you know, as the model gets more complex and things like that, it can be a very time-consuming and interesting process. It can take up lots of computing time. Many hours on the part of Salvador, who's done a really incredible job, like fine-tuning this process. And so for complicated models like this, you know, approach that we have adopted and also was similarly adopted by the Allen Institute's V1 model is that it might be necessary to specify layer-specific gain parameters. So say the gain, you know, excitatory to excitatory gain in layers 2 or 3 might be different than it is in layer 4. And so, you know, increasing these parameters does come with a risk. You know, increasing the number of parameters increases the size of the parameter space that you're exploring, and that increases the number of optimization trials, and, you know, that increases the risk of, say, getting stuck in a local minima where it's not, you know, exactly what you want, but you're kind of stuck in this part of the space. And so there's kind of two main ways to reduce that parameter space. So you can reduce the number of parameters or reduce the range of the parameter values. And so the way that, you know, that we've come up with, again, that also the Allen Institute used for the V1 model is to go layer by layer, as I mentioned. So parameters, you know, are first optimized in just layer 4. And so once this layer reaches kind of valid firing rates in all the cell populations, you add in layer 3. And the layer 3 connectivity parameters are tuned. And then, you know, things are kept within a small range of the previously obtained solution. So this is repeated, you know, for layer 2, for layer 5A and B, layer 6, and then layer 1. So yeah, so I just want to mention that, you know, as things get more complex, it might be necessary, and, you know, it might not be, but you may run into cases where it might be necessary to reduce the complexity of your optimization in this kind of bootstrap, you know, step-by-step manner. And so we're doing this currently using Optuna, which is a hyperparameter optimization framework. And here's a link here if anybody's interested in reading more about that. And just some more visualizations of this. So, you know, there's various ways to kind of look at visually to see what's going on. So, for example, here, you know, we have our fitness, and then we have different parameter values for the values that are down here. So you can kind of look at how the fitness stacks up for different values of a particular parameter. And here, you know, we have where color refers to firing rate of a given population. I believe these are cortical thalamic cells in one of the optimizations. So on the bottom here, we have the excitatory to excitatory gain. And up here we have EI gain, and then we've collapsed these, the inhibitory to inhibitory, inhibitory to excitatory. And again, we can see that for any given square within the fully connected network driven by background activity, for these, for this sort of triangulated gain parameter situation, this is the firing rate of that particular cell population using this particular combination of, network level gain parameters. So you can see there's a lot of different things going on, and it can be a very time-consuming process that takes a lot of computing time, as has been the case with us. So just wanted to mention that. And then, okay, so we can move on to kind of any questions on that. And again, I'm happy to provide more reading material or anything like that if people are interested in, any more of the nitty gritty of how this works. And feel free to message me on Slack also. Alrighty, so we can move on to kind of the last part of this step that I'll touch on, is that we then want to validate the model using empirical data. And this for us means looking at this under 3 conditions, which is what we're going to do. The first is going to be, we're looking, and this is what we're doing now, really, looking at the data at rest, again, both from the model and the monkeys. And the next thing we're going to do is be looking at the responses to click stimuli, and then finally to speech. So we're kind of gradually upping the complexity here, and seeing how things go. So for example, one of the things we'll want to look at is local field potential data. I know that there's been some exercises and tutorials and stuff where you guys have gone over how to record this. So in vivo, of course, the local field potential is recorded extracellularly. It's recorded in awake-behaving macaque monkeys. And then in our model, again, I believe you guys have gone over how to record LFP data. So it's calculated, it's the sum of the extracellular potential from each segment. And then for the estimation of the extracellular potential, we use the line source approximation method. And again, I'm happy to provide citations or reading material if anybody is interested in the details of that. And for consistency with our macaques, we virtually spaced our electrodes at 100 micrometer intervals along the vertical axis of the 2000 micrometer A1 column. So we follow the same recording protocol that they do in the macaques as well, to just increase the validity wherever we can. So here's an example of some of that from half-size version of this model. Again, feel free to ask me or some of the other tutors, or look at the NetPyNE documentation to see how each of these are generated. We have the time series, spectrogram, PSD. So they're just essentially different options on the record LFP function. And then also, one of the things we really want to look at is the current source density. So that's the second spatial derivative of the LFP, which is what makes part of what makes the LFP data so useful to look at. And this is very helpful because it tells us kind of the direction of current flow. What layer does it start in? Where is it flowing to, etc. And so this is just an example of some preliminary analyses that we've done using current source density in the spontaneous condition. So this is at rest. It's not being given any speech inputs or click inputs or anything like that. So just observing the pattern of activity in the supergranular, granular, and infragranular layers. Just wanting to eyeball this first and see if we can get a sense of whether the activity looks biologically plausible or kind of way off. And for now, the conclusion is that we're getting there. The activity we're seeing in terms of source sync activity and oscillation events has some similarities, but there's some differences that we're in the process of refining. And I'll show you another. Oh, it's going to load. OK, and this is similar to the slide I just showed, but this is in response to a speech stimulus, which you can see occurs at this kind of dotted line with this red triangle. So this is preliminary. Again, we're still at the step where we're refining the validity of the model in the spontaneous context, but just kind of proof of concept shows that the activity that occurs in response to a speech signal, again with this arrow dotted line, is approaching biological realism, which is important. And another piece of data that we want to look at besides current source density, this isn't speech, is also going to be the oscillation events. And this kind of gets back to the meat of what we were really looking for with this model. So we want to know what oscillations events occur, what frequency, where they are occurring, and what layers, what do they look like, so their morphology, their location. And to do this, we're using that event package that I mentioned earlier. It's essentially the same functions, the same software that's used to derive the information from non-human primates. We use that on the data from the model as well. Again, just, kind of detecting those oscillation events from recording data. And so just want to show an example of that as well. So on the left here, we can see an excerpt from simulation. On the right, we can see an excerpt from a macaque recording. So these are both in the granular layer of the A1 column. Up top, we see a spectrogram with frequency here and time here. And the red bounding box indicates the presence of a multi-cycle oscillation event. We can see this more clearly in the bottom panels here, which show the raw and filtered current source density signal over the same period of time. So this is an oscillatory event in the alpha frequency band, and one that kind of matches in terms of how it looks, the number of cycles, the location, across the monkey in the simulation. So ideally, what we want to see is that the monkeys in the model have a similar breakdown of oscillation events in alpha, theta, delta, etc. That's the kind of data that we're looking to see now. Let's see. So kind of looking to the future here, just wanted to mention, we talked about that first thing, but there's the second thing, which is, that entrainment relies on this phase reset mechanism that's orchestrated by these, thalamic matrix pathway neurons and these supragranular cortical interneurons. So how are we going to do that, basically? So essentially, this is the great thing about having a model is that we can go and we can really manipulate things in a much more easy way than we can, if this was a living system. So, can manipulate the circuitry, between the matrix pathway, between the matrix neurons and the interneurons to examine the effect on oscillatory entrainment. Does it disrupt entrainment? Does it modulate it in some way? And so we could, again, this is just a reminder of what that circuitry looks like. So, we could lesion the thalamic matrix, so to speak, set connection probabilities or something to 0. We could lower the connection strength between them. So instead of just outright lesioning, we could lower things, see what point do things start to change? What does that look like? We could also lower the connection strength up here between the, some of these supragranular interneurons and then some of the excitatory cells. So you can see if you have a model, and you've instantiated in this really organized way that NetPyNE allows you to do, it makes it very convenient to go through and be able to manipulate different parts of a circuit that you're looking at and see, what is the effect. And then, from there, we want to investigate the mechanisms of the thalamic matrix activity change during phase reset and entrainment. So we mentioned that briefly when I went over that kind of that diagram with the burst to tonic firing. And so basically we want to think about, as we're looking to the future, how do these phenomena help with segregation of attended auditory streams, versus background? Do these phenomena have any role in parsing the contents of an auditory stream? And, can we, say, devise a circuitry-based explanation for a question that I think is interesting, which is, why is stream segregation, in terms of I'm listening to my friend versus I'm listening to, the lecture that's happening, that requires attention, while parsing, appears to be this automatic sort of process that just happens without us thinking about it. If they both rely on this sort of, oscillatory thing, why does one require attention and one is automatic? Is there a circuitry-based explanation for why that might be? So just want to give a kind of broad overview of what this project is, the kind of questions we're looking at, and then sort of, looking to the future, what kinds of things might we ask, once we put everything together. So with that, just want to thank everybody. So Sam Niemoten and Salvador, who've been working with on the model, really fantastic. Bill, of course, our collaborators at NKI with Dr. Peter Lucados, obviously our neuron crew, Dr. Hines and Dr. McDougall, and along with, some of the other people at Downstate that have been, Dr. Stewart and QB and Dr. Freeman, that have been really great, our funding sources, of course. So thank you very much, and I welcome any questions on this, either here or on Slack.
Thank you so much, Erica. Fantastic talk. And I think Bill has a question.
Erica, so since you have the subcellular projection localization from S. crackum, and you have subcellular electrophysiological information from current source density, why not just directly, not that it's likely to work, but try to directly use the current densities as settings for the individual projections, or sets of projections, more likely.
I really don't know the answer to that question, Bill. I'll have to think about it.
Well, it'd just be something to try if you haven't tried it. I mean, I imagined you probably tried it and it didn't work. One of the problems will be then you have to set up a set of projections to a particular layer location and call that your parameter for manipulating instead of setting them as. So actually, it wouldn't be much different than what you do now.
So actually for A1, Macaque A1, we don't really have any S-Kraken data. It's all for
Right, it's all based on S2. I'm just saying it's the best you have, and since you have corresponding anatomical and electrophysiological, well, not that good correspondence because one's a mouse. I think the bigger difference is between the mouse and the macaque than it is between S2 and A1, or S1 and A1.
Yeah, that's probably true. Alrighty. Any other questions? Again, feel free to message me on Slack. If anybody asked questions and wanted me to give, further reading material or citations, just message me to remind me again so I know who asked, and I'm happy to send, anything to help you guys out with that.
Erika, sorry, I have another question.
Yeah.
So I don't know if you got already some information about that from the model, but I was wondering whether you could predict if there is some population, and I imagine that would be an interneurons population. If you have one of these that plays a major role in in the information processing coming from external input, because I saw that in your model you included somatostatin interneurons, paralbumin interneurons, and so on. So I was wondering whether so far you could predict if one of these population plays a major role.
Yeah, I mean, I think that, each of them definitely play a role that is very important. As I mentioned, the local interneurons, say in thalamus, are going to have a big impact on how, the thalamic signal processing occurs and, and how that's getting propagated up to cortex. And stuff. So I would say, for sure, the thalamic interneuron, the local thalamic interneurons are very important. Again, the supragranular interneurons, up in layers 1, 2, 3, also very important in terms of looking at this sort of matrix pathway, things like that. Salvador, I don't know if you have more to add on the different interneuron populations, but yeah, for sure, I don't know if in terms of just turning certain interneuron populations on and off, things like that. We haven't delved as much into that, but in terms of, what I would say are probably significant interneuron populations, the ones I mentioned,
Okay.
Yeah, I think it's really interesting to look into this. It's the first model that we really have all these 4 populations. Before we only had the PV and SOM. But we do have experimental papers suggesting like a specific role for VIP cells, for example. So I think it's quite interesting to look at it in the model.
Yeah, no, I was just wondering because, again, so I think it's clear that I'm a lot more into the visual system. But if I'm not wrong, I remember a paper where people were modeling the visual cortex. I don't remember, I gotta say, which layer, but they included in the model both the somatostatin and paralbumin interneurons, and shutting down just the somatostatin, they saw that there was a very big change in information processing, and the same didn't happen when shutting down the paralbumin, for example, interneurons. So I was wondering whether you observed something similar already in your model, but yeah, I understand that there is something that is going on still now. Yeah. Okay, thanks.
Yeah, of course.
There's a very interesting paper on CA1, a very large-scale model of CA1 by Marianne Desire in eLife. I can share the link now. And that includes 7 different types of interneurons. And she looked at each one at the role they had on the circuit. And it was very nice because she showed like some of them didn't have such a big role, and others played a major role in generating gamma oscillations and things like this. I'll share the link in a bit. Okay, so if we don't have any more questions, let's thank Erika again for her fantastic talk.
Yeah, I think Marco.
I have a question. I'm thinking So if you see like the oscillation of the audio is like similar to the global population oscillation of activity, I guess that could mean that there are straightforward correlation between this high frequency, high amplitude, and high population activity, and probably there are some sort of computer between single neurons that are trying to extract more information, something like that. But and that could explain this sort of correlation between the audio oscillation and neural population oscillations, because well, I think that the computation in the brain is like more single cell, and there should be a single cell explanation of these events.
Oh, so there are definitely, single cell. There are definitely things occurring at the single cell level, which are very important for this phenomenon. I would say, there's, if you look at inferior colliculus, as I was mentioning briefly, there are some cells that are tuned to, specific modulations that occur in the amplitude or the frequency spectrum. So there's definitely single cell responses that kind of, in aggregate make a very, big impact on the group, on the larger group response to and the larger group effect of, entrainment and things like that. It's definitely very, very important with single cell activity for sure.
Thank you.
Okay, so thank you so much, Erica.
Thank you guys for listening.
Thank you.
And we'll see you all guys tomorrow morning for the next.
Thank you.
Thank you.
Thank you. Bye.
Thank you. Great talk.
Thank you.
Thank you.
Alrighty.
Okay. Great. I'm giving you a lot of work with the cursor.
I love it. It's great.
Yeah, thanks so much. Really nice.
All right. Thanks, guys.
Okay. See you.